Good afternoon to everyone, and thank you for registering to this live webinar. My name is Fidel Tugizimana from the University of Johannesburg in South Africa, and I will be hosting this webinar on behalf of the Early Career Members Network of the Metabolomics Society. This is actually the 7th of our webinar series, and it is brought to you by the EMN committee. Currently, the webinars are free for all to attend by the courtesy of the Metabolomics Society, and I would like to inform you that all material will be recorded and uploaded to the Metabolomics Society website, allowing the access to the live content at your own convenience. Please connect with the Metabolomics Society on Facebook and LinkedIn as shown on the screen. I would like also to ask for feedback about the Early Career Network. You can share your ideas and suggestions at the email address on the screen. Before introducing our speaker today, I would like to remind you that the International Conference of the Metabolomics Society, Metabolomics 2016, will be held in Dublin at the end of June. You can join and visit the Metabolomics website. And the EMN committee is hosting a welcome social mixer on Sunday 26th, 2 workshops on Monday 27th, a reception on Tuesday 28th, and it will be sharing Young Research Scientist talk, which will be on Wednesday 29th. Please join us, and all this information, you can find them on the conference website. And at the end of this session, I will give more details on the 2 workshops that are organized by the EMN. Today, our expert speaker is Dr. Karl Burgess. He is a bacteriology research fellow in the Institute of Infection, Immunity and Inflammation at the University of Glasgow, UK. His research interests focus on the use of mass spectrometry in biomedical research, particularly in the understanding of infectious disease. He is the head of the Metabolomics facility, which is part of Glasgow Polyomics. He is a Wellcome Trust Fellow studying the mechanisms of adherence in pathogenic biofilms. There will be time after the webinar for questions, so please send your questions by using the questions option of your webinar application. We can type the questions throughout the presentation, and our speaker will answer them live at the end of this webinar. I think we're ready to start. I will pass you on to our webinar speaker, Dr. Karl Burgess. Thank you.
Thanks very much, Fidel. I'd just like to start off by saying that it's a real privilege to be here and, well, to be in my office giving this talk to you guys. And it's a great opportunity to talk about some of the research we're doing. It's kind of a controversial title, Chromatography and Metabolomics, what's the point? And I just wanted to start off by making it clear that I'm asking kind of what the point of chromatography and metabolomics is. So hopefully I might be able to answer that question at some point during the talk. So I pulled the definition of the metabolome off Wikipedia while I was writing this talk, and it's important to bear this in mind, I think, every time I'm trying to design an experiment or something like that, to go back to the overall definition of what the metabolome is, because the metabolome refers to the complete small molecule, complete complement of small molecule chemicals found within a biological sample. And, okay, the biological sample can be all sorts of different things, but when we're doing metabolomics, the kind of principle behind it is to do the whole metabolome. And we don't do that, and I don't think anybody will ever be able to do that, to analyze the complete metabolome in one go, in the same way that nobody's going to be able to analyze the complete proteome in one go. And so metabolomics not only has lots and lots of different names now, people call it metabolomics, metabonomics, phenomics, but also because of this kind of complexity of the system, and because of the difficulty of analyzing all these different molecules, we tend to subdivide it based on the type of molecules. So we've got lipidomics and glycomics and things like that, but also the kind of applications that we're doing with the science, so fluxomics and things like that. But I think what's important to bear in mind is that overall all we're doing is analytical chemistry on biological molecules. And I think this is more obvious in metabolomics than it is in any of the other omics. And so if you look on the right side of the screen, it's kind of hard for me to gesture on this, but the analysis of the metabolome is really supported and kind of dragged out of the ground by all of these different technologies that people use at different times for different molecules. And these comprise all sorts of different things. You've got different detection methods, Raman, FTIR, mass spectrometry, NMR. You've got different separation techniques, gas chromatography, liquid chromatography, capillary electrophoresis, and the fractionation things that you might do at the beginning, up front, before you prepare your samples, are also a kind of separation technology, solid phase extraction, solid and SPME and things. And even the ionization sources that you use, if you're doing mass spectrometry, have a dramatic effect on the type of things that you'll see. So using electrospray or DESI or SIMS, something like that, will give you lots and lots of different types of information. But I think what's really, really important is to bear in mind that the thing that we're always trying to analyze is the biology, is metabolism. And this is where the experimental design comes in. So part of what I was trying to get across with the lots of different methods for doing metabolomics and this idea that we can never really do metabolomics is that every technique we use, every type of sample analysis, everything we do produces bias. And this is just something that we have to live with, and it's something we have to account for and deal with as scientists. And so sample selection is the first point at which bias is introduced. And recently, during a course that I was involved in with Reza Salik and Ron Behrens, Reza and Ron described a situation where they were doing analysis of apples from the same tree, and they found that if you take an apple from one side of the tree, its metabolome is quite substantially different from the metabolome of an apple from the other side of the tree. Now, intuitively, I would think that that was just a technical replicate. It's the same organism, it's a different part of the same organism, ideally the same kind of organ. And so ideally you'd think that was a technical replica, but actually you can get these significant changes just based on these confounding variables, the position, gender, hospital the samples were taken from, the collection method used, all of this type of stuff. The type of experiment that you design has a dramatic effect on what you'll then see. Obviously then, sample preparation is the next massive thing that you have to bear in mind, because the type of extraction methodologies you use will dramatically affect the type of metabolites you pick up, especially with LCMS. Your separation, your extraction solvents, you can either dry things down and then resuspend and inject, but you'll lose things during the dry down. Things will stick to the sides of the tube. The solvents that you're using have to be compatible with the chromatography system and the mass spectrometer, and I'll talk a little bit about that later. But this also introduces significant bias, because the stuff that you leave in the bottom of the tube, the debris, will probably contain some small molecules that you just aren't extracting appropriately. The next thing is sample storage and transport. And this introduces bias. It also introduces destroyed experiments in many cases. And as somebody who runs a core facility where we take samples from lots of different countries, some of these poor guys will spend months and months preparing samples, getting the extractions right, getting everything absolutely perfect. And then we found that people were tending to ship on dry ice international with tubes labeled in magic marker 1, 2, 3, and 4, just loose in a box full of dry ice. When they'd arrived, the top had popped off and we'd lost most of the samples, and the chloroform and things like that, and the solvents would have just wiped off the numbers. So that can totally destroy experiments. But what temperature you ship the samples on, how long they've been left in transit, and things like that will also bias your samples. Another thing that I think people don't bear in mind a lot is that when we're injecting samples, they don't come straight out of the freezer into the instrument. We generally set them up on a plate, and then things get run, and it takes hours and hours to get the last sample on. So you'll get effects due to sample storage, you'll get bias due to sample storage, and So people like Dave Broadhurst, Ron Varens, we've produced batch correction algorithms that will deal with drift and the degradation and things like that, and the degradation of the instrument during these batch analyses. And if you're doing really large sample sets, I'd really suggest that you start looking into methods that help you deal with these kind of systematic effects. Obviously, which column you use if you're doing some kind of chromatography method is going to massively bias your detection system, what things you'll pick up, and how ion suppression affects things. And obviously, the type of detector that you're using has a dramatic effect. No detector is universal, and some things are more sensitive than others. So my biggest interest is in mass spectrometry. Mass spectrometry can only detect ions. So if it's not an ion, you won't see it. NMR is much broader and will detect a signal because you tend to be looking at protons or C13 atoms. But the signals tend to be lower, so you're looking at maybe 20 to 30 metabolites from a single one-dimensional analysis. So there's all sorts of different biases and different effects based on what the systematics that you've got in place to do your experiments consist of. And so this is something that's really important to bear in mind when you're doing a metabolomic analysis, because if you think you're going to be able to analyze the whole metabolome, you're in for a big challenge. So I'm just going to go through the basic concept of how chromatography works. I'm sure all of you or most of you already know. So in liquid chromatography, you have what's called a column, which is basically a tube. It's packed with beads, which tend to be described as the support. And the support is coated with something called the stationary phase. And the stationary phase is called that because it doesn't move. And the stationary phase is some kind of chemistry. The canonical one for most applications is C18, which is a kind of aliphatic carbon chain. And this is quite hydrophobic, and so it's good for sticking fatty substances and things like that. You can also coat it with all kinds of other things and have different specificities. And this is one of the great things that makes chromatography so powerful, is that you can have different chemistries that allow you to separate different things, different analytes with different specificities. And so, of course, the next major component of this is the analyte. And the analyte is introduced to the column using the mobile phase. And the mobile phase is called that because it moves. So this can be a gas if you're doing gas chromatography, or a liquid if you're doing liquid chromatography. And it's just whatever solvent system that you're using in this to introduce your sample to the column. And this is the kind of idea that things that stick to the stationary phase will go through slower because they'll interact more with the surface, and things that are less sticky will go through quicker. And so at the bottom right you can see this little kind of cartoon of the blue peak coming off before the red peak, because the blue goes through faster. And that's basically the principles of chromatography. Gas chromatography is exactly the same as this, except that instead of a packed tube, you are generally dealing with a tube where the surface where the stationary phase is coated onto the wall, and therefore the columns tend to be much longer, and obviously that you're using gas as a mobile phase instead of liquid. And one of the things that I think we have a lot of problems with actually in metabolomics is and especially chromatography, liquid chromatography, is this concept of ion pairing reagents. Ion pairing reagents are something that are used a lot in chromatography, in good chromatography experiments, and they're kind of adapter molecules. And so if you want to improve your chromatography for, say, polar species or something like that, and you have a C18 column, you can improve the chromatography by adding an ion pairing reagent. And an ion pairing reagent is something that maybe has a hydrophobic part and a hydrophilic part. So it will interact with your, say, C18 column, and kind of stick to it a bit, and then it will provide a modified surface that can then interact with your polar analytes. And so this is a kind of common technique for getting improved resolution and tweaking the chromatography specificity of a standard column to get the result that you want. The problem with this is that nearly all of the really good ones are mass spec incompatible, and actually in the list here, none of these are really ion pairing reagents. They're really just buffers, not even particularly good buffers at that, because they don't have the size to be kind of amphipathic. So the main problem with this is that if I'm talking to a chromatography company and I say we really need a better separation system for dealing with the problem of the metabolome, and they say, well, what do you want to pick up? And I say, well, everything. Fatty acids, amines, phosphosugars, etc., 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 etc. And I go, okay, and they have a think about it for a bit, and then they'll say, well, what about this column? And I go, okay, well, what ion pairing reagent are you going to be using for this? And they'll suggest some kind of horrific thing that you cannot use anywhere near a mass spec. And this is the kind of example of what happens to your mass spec if you put the wrong solvents down it. This is something that you don't ever want to have happen to your mass spec, and it'll take you a long time to clean out. So we're kind of hampered in a sense in that we have to use the chemistry that we've got that's actually part of the system to do the experiments that we want. And so this is an example. So one of my PhD students, Lawrence, has just finished up, and one of the things that he had to do in his corrections was provide some more information about the different column types that you could use in metabolomics. And he said he was looking at this thing from the Chromatography website as a basis for a diagram, and it just gives you an idea of how kind of horrifically complicated metabolomics is. If your target compound is water soluble or fat soluble, you do a different thing. If you then, depending on what type of compound it is, if it is water soluble, you then have to subdivide that into various different categories and things. And so, and each one of these has a different type of column associated with it. And some of these columns have solvent systems which aren't really mass spec compatible as well. So in metabolomics, we're really kind of, LCMS especially, we're really kind of pushing at an area that's very, very difficult to develop. So we can push fairly hard on the chromatography side and really improve things by going to high-resolution gas chromatography or something like that. And we were privileged to get a GC Q Exactive in the lab for a while. Which is the instrument on the left in this picture. We've got far too many instruments stuffed in a fairly small room. And this is, so Stefan in my lab has done a lot of this. And we really like this instrument because the resolution of the chromatography is really fantastic, and we can get sort of 2 or 3 second peak widths. And we're talking about maybe 100,000 theoretical plates, which is a kind of measure, an overall measure of separation power. When our standard Helix system has a separating power of about 5,000, so we're talking about a 20-fold resolution difference, and it's really, really useful for getting separation of all these things like phosphosugars and sugars and things that normally are really, really difficult to separate using LC and produce lots of different peaks. But the drawback is that you are using electron impact ionization, so there's some molecules you won't see, and you don't tend to pick up quite as many compounds as you do with LC, so there's always trade-offs. You're always biasing your analysis in some way. So to go back to LC, we tried to put together the kind of broadest possible separation that we could manage, and this is a 2-pump system that we published, I think, last year or the end of the year before, where you're using a C18 column to get your hydrophobic separations, and we inject the sample effectively onto that using 1 pump, a very low flow rate for kind of normal chromatography of 65 microliters a minute. Anything that sticks to that column will stick to it. Anything that doesn't stick to that column flows straight through the column, through this T-piece, because you've got another pump that's pumping through, and hits this Hillock column. Now normally this is really, really hard to do because the gradient that you use on a Hillock column and a C18 column are basically opposite. You are using on a C18 column, water is a weak, strong solvent and acetonitrile is a strong solvent. On a Hillock column, water is a strong solvent and acetonitrile is a weak solvent. So normally you just wash stuff through. So that's why we went for this 2-pump system. So what's really important here is the dilution effect. So in pump 1 you're only running at 65 microliters a minute. At pump 2 you're running at 300, 350 microliters a minute. And no matter what solvent gets through the C18 system, whatever composition of solvent, whatever gradient you've got running on it, by the time it hits this T-piece, it's going to be diluted so much by the 350 microliters a minute that we're running that at, that it's not going to really affect the retention on the Hillock column. And this works pretty well. So It's a single separation. We just run both gradients simultaneously on the two different columns. We inject one sample, and we can see from this that if it's a compound that would normally not stick at all to a reversed phase column, but would stick very, very strongly to a helic column, it tends to stick strongly to the helic column, to the combined column. Conversely, if we're looking at things like bile acids, which don't stick very well to helic, but stick pretty well to reversed phase, we get pretty good retention of those. So it's kind of a broad-based analytical method that works for a lot of systems. And this is just a kind of real-world example with a complex metabolomic sample. But even then, there's some peak tailing, we don't detect everything, and there's limitations to the mass spectrometer that mean that, you know, if we're trying to detect these really small molecules, then we maybe miss a bunch of the lipids and the larger metabolites and things like that. So we're still dealing with a biased system. And when I sort of think back on the methodologies that we've developed and the stuff that we've done over the last few years, we've had a lot of successes from the untargeted metabolomics, and it's been a really, really powerful technique that's very well established now for generating hypotheses. But I think some of the most satisfying experiments that we've done are ones where we use something very, very simple and narrow. And this is an example, so we got this published in the ISME journal recently. And actually, the whole experiment ignored all of these compounds apart from serine. So all we did was an experiment that very nicely separated the chiral enantiomers of serine, the L forms and the D forms. And we could get some quantitative information out of the different ratios, and we were looking at the different niches in E. coli and which carbon sources they consumed and things like that. So it's provided a bunch of really interesting data from what to me is a very, very simple experiment. One compound, one method, you're using an extracted, basically high-resolution extracted ion chromatogram to look at a single molecule, separated into two peaks based on its chiral structure. And this is not really metabolomics. This is an analytical chemistry application. I don't think anybody would try and convince me, would want to try and convince me that this analyzes the metabolome in any way. And if you're really only interested in measuring glucose, then your best instrument is probably going to be a glucose testing meter. So it's one of these things. It's about experimental design and what you do with it. And so because of this and because of these kind of broad-based metabolomics experiments that we were doing, the different breadths of different chemicals that we pick up, you get lots and lots of different qualities of peaks. Some of them are good, some of them are nice kind of Gaussian things. Some of them are bad, they're jagged, they're not well resolved, they smear off the column, they don't even form columns that you get shoulders all over the place. And some of them are just kind of, you're not sure whether they're background noise, chemical noise, or what they are. So we've got the good, the bad, and the ugly of chromatography. And I started to get really kind of jaded about the whole idea of chromatography and started to look at alternatives. And then I thought about this weird problem that we had ages ago with our instruments. And if you look at this top panel, you can see that what looks like a reasonably Gaussian peak. And then if you look at the M over Z as it goes up, you can see this other one start at 118.147, and it kind of goes up, and then it kind of goes down, and oh wait, it's gone back down to 118.138. So yeah, and then if you look at the one below it, this peak seems to do a kind of weird loop-the-loop and disappear off the bottom of the detector. And so mass spectrometers are usually pretty reliable with these kind of things. They produce the same sort of peaks over and over again. But every now and then you'll get some weird electrical noise, some kind of processing problem, some kind of issue with your data. And actually, this is common to every kind of data collection. You really need to kind of have a look at the samples that you're looking at, the data that you're getting. You need to be critical, I think, and look at multiple methods, look at everything you can to get the best picture of the story that you can. So chromatography might have its problems, but I think any analytical method has its problems. But one of the things I'm really interested in at the moment is that the whole focus on chromatography is to try and get more data. In proteomics, for example, you routinely do these massive two-dimensional LC experiments where you spend maybe a whole week doing a single experiment, and then you flush the system out and start again for the first replicate the next week. And this is just to get the biggest possible coverage that you'd possibly manage. And maybe started thinking about it from another perspective, and one of the things that the computer scientists and the statisticians are always saying to us is, why do we measure so many things but so few replicates of them, such small numbers of them? And I started to think about this, and maybe this is the next way to go. So one of my students, his project was to build this desorption electrospray imaging source, which looked quite tricky, but it seems to work. So this is pretty cool. So I'll kind of show you how it works. It's basically the same as an electrospray source. It was invented in the Cook's lab quite a few years ago now. And you've got the standard sort of setup. If you look at the little diagram on the side, you've got your solvent flow going through a needle, you've got your nebulizing gas, and you've got your electric potential on it, which generates droplets and the spray. But instead of putting your sample into the solvent flow and spraying it directly into the mass spec, what you're doing is putting nothing in the solvent flow and basically bouncing the spray off a surface. And if you can see this kind of red dot, which is getting stuff desorbed from it into the little droplets that are then going into the mass spec inlet. Producing a DESI source is actually relatively straightforward. You can kind of we took an old Proxion stage that we were originally using for nanospray and just kind of modified it. We built a little sprayer using a capillary and some stainless steel tubing, so just a few bog standard fused silicon capillary and some stainless steel tubing. And if you get the angles right and a decent surface, you can get a DESI spray. It's relatively straightforward, and there's some fairly good papers out that go through the exact process of how to do it. But we wanted to image with this. We wanted to get spatial information out of this source. And so we built an XY stage. And so most of it's built out of acrylic, just cut using a laser cutter. It's just got 2 stepper motors and one of those little Arduino microcontroller boards and the software to run it. And it's a simple program. It basically just turns the stepper motors and scans from left to right and then moves down, scans from left to right and then moves down. And this works pretty nicely. And so here you can see the setup that we use, and we're actually using this really not from a metabolomics point of view, but actually from a document analysis point of view. But given that most of the small molecules and things like that are in old inks or from biological sources, I think we can get away with it. Anyway, so the tube on the right is the thing that the spray comes through, and you can see that we've set it to this particular. There's a specific distance you need from the paper, and between the inlet of the mass spectrometer, which is the kind of bent tube on the left-hand side, and the angles, all the distances and things like that are really critical to get the right spray out of it. And there's this beautiful paper in JAMS a few years ago where you can determine based on where they went forward and determine based on the impact angle and the spray to surface distance and things like that, what kind of compounds that they could pick up. And this just beautifully goes back to the point earlier about bias. I mean, just the angle that you spray at determines the type of metabolites that you pick up using DESI. And so you need to design your experiment. You need to work out what you're looking for and optimize for that sort of situation to get the best out of it. But once you've done that, and once you've got something that works, everybody doing DESI starts off with rhodamine dye, which is the one that you get in magic marker. And so we've just written polyomics in a magic marker, overlaid the image on the mass spec image, and we've picked out that particular mass. And we can see that although it's kind of a bit boxy, we can get a fairly good matching of the image to the mass. And then the one below is a different image, and this is a kind of real-world example where we're looking at a bunch of different inks. So these are 8 different inks, and the 2 that are lit up in red are both derived from ivory. They're both called ivory black, so they've got some carbonized ivory in them. and some other chemicals, some acid and stale beer. And we can pick out particular molecules that are unique to ivory black, and the idea is that we can use this to fingerprint different inks. But obviously you can use this to image and produce two-dimensional data or three-dimensional data, including mass information out of a whole range of different compounds. It doesn't work for everything, but it's a useful technique because you don't have to do very much sample prep at all. So that's space. The other thing I'm really interested in is time. So Uwe Sauer wrote a paper, published this paper in Nature Methods last year with this beautiful technique that uses a couple of pumps and a fairly interesting setup. I'll just kind of go through it a bit, where you've got a cultivation vessel, which can be, I don't know, any kind of flask or tube that you're growing stuff in. You've got a peristaltic pump, and you put both ends of the peristaltic pump basically into this culture vessel, and continuously cycle stuff through it. But you connect the system up to a 6-port valve with a loop in it. And over time, maybe every minute, you can switch that loop and take a sample from your growing culture and then take it through another loop, mix it with some mobile phase, and slug it straight into mass spectrometer. And you can see here at the bottom, you can get these beautiful curves of different compounds. In this particular case, he used yeast, where he starved yeast for a while and then dropped in some glucose. And you can see that there are dramatic changes in a few metabolites. And it's not going to pick up an enormous number of metabolites. But there's not really very many other techniques that can give you, minute to minute or even faster in some cases, analysis of what exactly is going on in a growing culture. Obviously we get the medium rather than the cells, but we get some really nice information out of it. So this was my first attempt to have a go at it. So all I did was I got some glucose solution and dropped some baker's yeast into it. And you can see that valine is massively increasing and then dropping back down again. These samples were taken every minute. And so we're looking at the course of about an hour and a half. And there's a couple of tetrapeptides and things like that are appearing and disappearing. And these are very, very putative identifications. It's worth bearing that in mind. And we can do the same with glucose and see glucose dropping if we switch into negative mode. The problem with this is that we're always identifying things when you were trying to do these direct slugs. So at the moment we're using mass, and we're very focused on using mass in mass spectrometry because that's mostly what we get. So chromatography basically gives you a dimension of separation that narrows, that reduces the complexity, removes some problems with ion suppression, things like that. But what it really does is it provides a dimension of physicochemical properties that narrows down the complexity of your system, which means that you can identify things with greater confidence. So there's many, many methods for doing that. And many of them can be hyphenated to mass spectrometry. The most obvious one, the one that comes on virtually every instrument nowadays, is fragmentation. And again, I'm guessing that most of you know how it works, but a very simple kind of cartoony idea of just in case you don't, is that you have your analyte, and in collision-induced dissociation, CID or CAD, collisionally activated dissociation, they're basically the same thing, is available on most instruments now. It's the kind of standard way of fragmenting things. You take your analyte, you pass it into a chamber containing an inert gas at a certain temperature, and the analytes collide with that inert gas and produce fragments. It's really very, very simple. And those fragments, their sizes depend, they're partially random, but they're also directed by the structure of the compound. So what you end up is a distribution of peaks from a single peak. And if you look at this system, you can isolate a precursor ion, you can get a bunch of fragments that are associated with that produce a list of masses that can tell you some things and give you some more information, give you some structure of your compound. And some modern instruments, Orbitraps, and I think the Shimadzu ion trap TOFs, for example, can do these multiple rounds of fragmentation. So you get MS to the 2 and MS to the 3 and MS to the 4. So we can select each one of these MS to the 2 fragments and fragment them again to get yet more information out of a single run. And some of these instruments are really, really fast now, so that you can do this on pretty quick timescales. And we can use this. We can exploit this. There are certain databases available now. NZ Cloud, for example, has ion tree fragmentation like this. MassBank is starting to build it in. Magma as well in the Netherlands supports this kind of fragmentation. And it's really a good way of analyzing things. But again, there's always going to be some compounds that it doesn't work very well on, that sometimes just don't distinguish very well between each other. So another way of doing it is ion mobility, and this is, I think, is a technique that's been around for a very long time, but I think it's reaching the potential at the moment to be quite powerful for adding another dimension of separation. So I like to think of an IMS system as basically a wind tunnel for ions. So you introduce your ions to a chamber, and you have a gas flow, and they're pulled along the gas flow by an electric current. And the ions with larger cross-sectional area will go slower than the ions that are small, have a lower cross-sectional area. And this is kind of the idea. You have your buffer flow going in, your exhausts are going out, so pushing this way, you ionize your samples, and you have a detector at the end, ion gates as well to sort of shut it off when you're not using it. Your ions kind of go to the detector. Recently we sent some samples to TOFWorks in Switzerland. We've got a mix of standards that have each different isomer separated into different groups, and we just wanted to see whether we could separate some of those. And certainly I'm not aware of many systems that can manage this. And the cool thing about this particular instrument is the resolution is very high. It's about 200 on the IMS cell. And so these are some examples of different sugars. We were unable to get any separation from the from hexose sugars at with just the H minus, sorry, the minus H plus minus, so the 179 peak. But with the sodiated version, the steric hindrance, the kind of bigger kind of area that you have from having a sodium attached to the molecule, was enough to actually distinguish these. So we can get shoulder peaks and some slight separation of these. So we're really getting to the point where we can separate sugar isomers and sugar phosphates and things like that by ion mobility. It's got a resolution about 200 to 300, so compared to GCMS with 100,000 resolution, it's not really there yet. But the key thing about this is the drift time. So if you look at the bottom on this on the next slide, we're looking at millisecond timescale, so 23 milliseconds, 50 milliseconds, and things like that for this separation of beta-alanine from L-alanine. And that's something that we can do very, very quickly, the same kind of timescale as fragmentation. And ideally, you'd want a system that could do both, where you get an extra dimension of drift time, you get an extra dimension of fragmentation. And that's where you really can start to push down the different kind of compounds that you're looking at. So I'm pretty much coming to the end. I think the key take-home message from this is that we can't really do metabolomics completely, and the only way to get the right answer to the experiment is experimental design, and to be very careful with what you're looking for, your confounding variables, to really think the whole thing through before you start. And that's a good place to start, and I think you're much more likely to get what you want out of an experiment if you spend an extra few days designing it than if you rush in. The next line is a quote from The Hitchhiker's Guide to the Galaxy, and it's one of my favorite ones, and it's where they decide that they're going to build a giant computer to work out the answer to the ultimate question of life, the universe, and everything. And the philosophers break in and they demand that this machine, they shouldn't have a machine that just tells you what the meaning of life is, because that's the job of the philosophers, and they want rigidly defined areas of doubt and uncertainty. And this is something that I want from metabolomics. My problem with metabolomics is in the identification world, in knowing what a particular ion corresponds to in terms of a compound, is how ambiguous is this ID? you know, if I pick up 178.0550, it's probably a 6-carbon sugar, but it might not be. There's a lot of different things that could have that particular mass. It depends on our mass accuracy. We could have different formulas and things like that. What I'd like is a metric that tells you how uncertain we are. What is it? 10%? Is it 20%? You know, how many different things could this be? And the final thing is, we spend a lot of time and effort trying to get more and more and more molecules. Is that what we really need to do? Do we just want more data points? Do we want to see the single metabolite and how it changes in space or in time with greater resolution? Is this not going to give us more information maybe than 20 more identifications? And I think that's just something to think about. And the idea of where I'm pushing into at the moment, research-wise, is this idea of doing metabolomics in time, in growing cultures, or doing it in space, straight from tissue samples, slices, or straight from different surfaces and things like that, and documents. So I'd like to thank everybody who's stayed on the call. The collaborators at Thermo Fisher, everybody in polyomics, people in computer science who've helped us out an awful lot, and engineering in Glasgow University. I'd also like to thank Fidel and Reza very kindly for inviting me to do this talk. And I'd also like to thank all of you guys for listening. Thank you very much. And I'll just pass you back to Fidel for a final word.
Thank you so much, Dr. Karl, for this wonderful presentation. And then it raises questions and put us to how do we do metabolomics, questions regarding chromatography, questions regarding identification. Thank you indeed. It's time for questions, but before we start the question session, as indicated earlier, just want to give you some more details on the 2 workshops that are organized by the EMN. The 2 workshops are career development workshop and just a moment, it will come on the screen. And career development workshop, and which will just touch those few points: how to effectively communicate science through journal publication or grant applications, how to establish scientific collaboration, career paths options after postdoc, and how easy it is to move between academia and industry. Please join us, and I'm sure you can gain more knowledge and insights and this practical aspect in a research life. The second workshop organized by the EMN is the importance of experimental design, data acquisition, and quality assurance in metabolomics. As Karl touched on it, and at the end, study design is really essential in metabolomic studies. And this workshop will touch on those aspects, source of experimental errors and the consequence of poor study design, experimental design strategies and how they can affect the outcome, data acquisition considerations and quality assurance measures, and the quality control assessments and data cleaning. If you are coming to the conference, please join us at those pre-conference workshops. I would like also to invite you to our next webinar series, which will be on the 27th May, same time, and it will be from feature to compounds, how to get started and move forward. The aspect that just Karl touched on, the identification of metabolites. Thank you again. Karl, we have some questions here. For you, and One question, with GCMS, why the non-confident ID list is not increasing as one would expect? Is the problem on mass spectrometry or annotation, or because the 2D GCMS and increased column length has not helped? What's your take on that?
I'm not 100% sure I understand the question, actually. So you're asking whether in comparison to LCMS or in comparison to
the GCMS, there's NIST libraries and many libraries that they, with the increase of two-dimensional GC, as you showed the chromatography, but the list of the IDs or annotation seems not to be increasing with that improvement in technology.
GC is a really, really interesting technology. It's something that we've been working on for a while, and yeah, there's a lot of things to think about with GC. It's an analytical technique like any other. So the NIST libraries, the Wiley libraries, and Golem and the various other ones are really, really nice to have. The only problem with it is that because it's mostly based around electron impact ionization, you tend to not see the molecular ion in general. So if you miss the molecular ion, you don't necessarily know what the intact mass of the compound is. And on LCMS with electrospray, we tend to have that. So that's a really, really good filter right from the beginning in terms of what things you're picking up. If you don't have that, you're basing your entire identification on the fragment pattern. And the fragment pattern can be somewhat ambiguous, depending on, I mean, fatty acids are a great example of this. They tend to have very similar fragmentation patterns, but their retention time changes based on chain length. You don't see much at the top end of the fragmentation as the chain length gets longer. And what you need to rely on is retention time. And that's where retention indices and things like that come in, which they also have databases of, because GC is really, really stable for this sort of stuff. But yeah, it's a really, really nice technique.
Okay, thanks, Karl. Another question. With this 2D chromatography, what, in your opinion, what is the future? Are we going to be adding more dimensional chromatography, or, as you said, it's difficult to separate the complex mixtures? To what extent?
I really hope we aren't going to go for more dimensions. My whole PhD was based around doing separation of intact proteins by liquid chromatography, and by the end of it I was doing two-dimensional protein separations followed by digestion, and then slugging each one to a one-dimensional LC peptide separation. And the experiments used to take about a month each, so it was an absolute nightmare. I think it's a difficult question. So for some applications, if you've got your own instrument and you've not got a huge number of samples and you want to really get as much information out of those samples as you possibly can, I think two-dimensional separations can be a really great idea. I think, though, a lot of the research, a lot of the technical development into metabolomics is driven by clinical analysis, and clinical analysis is really pushing larger and larger numbers of samples. And I know Jules Griffin is working on a direct infusion methodology for clinical samples just to get that throughput. You know, you need to push samples through very, very rapidly. And any time you do a separation, take fractions, and then have to re-inject all those fractions, you're exponentially increasing your runtime. So, and also your chances of something breaking. You know, if you've got one run per sample, then it probably work. If you've got 10 runs, you've got 10 times the chance that something's going to break during that analysis. So I don't miss it from my days of doing proteomics, of just doing 1D analyses.
Okay. Thanks, Karl. Another quick question. Based on your experience with chromatography, what are the best approaches to optimize the gradient for a particular column? Or what are the how do we assess the quality? Should you spend time trying to optimize, or really minimum separation is enough for metabolomics?
Again, I think it would depend. How much time have you got?
Yes.
If you need the results by tomorrow, then maybe just doing the fastest one you can possibly get will work. I think it's always worth spending some time trying to get the best data that you possibly can. And if you wouldn't be prepared to present the data at a conference, then you probably shouldn't try and do something with it. You know, you should always try and get the best you can. Another thing that's really important that I think this question kind of touches on is if you've got interference and things, if you're developing a targeted method or something like that, and you have isobaric or isomeric compounds that can interfere with your samples and things like that, it's very worth optimizing to get a retention time difference between those so that you're not getting any interference if you're trying to do quantitation or you're trying to get an identification. So for those purposes, I think it's absolutely worthwhile.
Okay.
Sorry.
I guess one of the last questions, what would be the best way to analyze carbohydrates in metabolomics?
Whichever one you've got. It's the simple answer. We've had quite a lot of, it depends on what you mean by carbohydrates. If you're talking about really like big ones, so polysaccharides and things like that, I've not got that much experience of it. Although using a lot of the techniques that that have been used for glycan research, you probably wouldn't necessarily even need to separate them nowadays. You can probably just fire them straight into the mass spec and use some kind of clever fragmentation stuff to break them open. If you're talking about sugars and things, ion chromatography is quite a nice way of doing it. So you can separate carbohydrates pretty nicely using those methods. And ICMS is kind of taking off a bit at the moment because it does give quite good separation of those. So that's probably what I'd recommend. But I think there's various columns for carbohydrates as well. There's Waters amide ones, which I think are pretty good, but I don't have a huge amount of experience with them.
Okay. Thanks, Karl. The last question, and I think it will be off. The isomers issues. Yes, to separate them, to annotate them, either on the side of chromatography or with the help of MS. Any input from your side, from your experience?
It's been said that identification is the most difficult question in metabolomics, and it's one that I pretty much agree with. It's actually a really hard thing to give any information out because it will very much depend on the analyte you're looking at and the particular isomers you're talking about. If it's something that has the same mass, the same formula, but a completely different chemical structure, it's probably pretty easy to determine it. And if it isn't, the physicochemical property that you use to differentiate it between will again depend on the compound. So the question before, how do you separate sugar isomers? Right? Maybe use ion chromatography. Maybe that's the best method because the fragmentation patterns are very similar. But maybe you've got a different system where chromatographically they all come off at the same peak, but IMS separates them perfectly. I think Edwin Madala had a beautiful example of that recently. And so it's a very tricky one. Getting a good identification is very hard, and convincing everybody that you've got the right one is pretty hard.
Yes. Maybe Jan will tell us more in our next webinar.
Yeah, ask him, not me.
Thank you so much, Karl, and I thank you all who are attending. I think it was really wonderful, and surely there's quite a lot of questions that one ask, and through our experience working in metabolomics, I'm sure answers will come. Thanks a lot, Karl.
Thank you.
Thank you, everyone, for attending.
you.